How do I do that? Do you know?
Just like mute the sound, sorry.
Okay, I'll try. Go ahead. If not, I'll close it if it like starts making funny sounds.
Okay. And I think we're good. So I will just give you the thumbs up at the. I think we can
like
go.
mute the sound, sorry.
Is it echoing or no? Are we okay?
Should be okay.
Oh, okay.
I think you can go, Liza.
Perfect. Welcome to our bi-monthly, our early Korean immunology seminar series. I'm really excited to introduce Maria Tokuyama, who is going to talk to us about endogenous retroviruses today. Maria did her BS work in biochemistry and molecular biology from UC Irvine. Then she traveled a little bit up the coast and did her PhD at UC Berkeley. And then actually moved all the way across country to my neck of the woods to Yale, where she did a postdoc in the Akiko Iwasaki's group, and then actually stayed on for several years as an associate research scientist. And I think this is where her love for endogenous retroviruses really started. She had a beautiful PNAS paper on ERV map, or mapping the endogenous retroviruses across the human genome. And since 2021, she's had her own group at University of British Columbia, where she's been incredibly successful and has received a number of awards, both sort of from the Canadian government as well as from the University of British Columbia. And we're really thrilled to welcome you here, Maria, and excited to hear all about crosstalk between endogenous retroviruses and immunity. Thanks so much.
Thank you. Thank you so much, Liza. Thanks for the introduction. And really, I'd first like to thank all the organizers of the Early Career Immunology series for giving me this opportunity to share some of our work here today. I think this is a really great way to connect with other ECRs and ECIs across countries and even continents. And I'm really looking forward to making new connections here. So before I begin, I'd like to acknowledge that I live and work on the ancestral and unceded territory of the Musqueam people. And I acknowledge that we in Canada have a long road of reconciliation ahead. And as I walk my own path of learning and decolonization as an uninvited settler, I'm striving to respect the vision and mission values of the peoples whose land we're on today. Okay, so really at the highest level, my lab is really interested in understanding the underlying basis of heterogeneity in health and disease. And really this is dependent on a complex interplay between immunological, environmental, hormonal, demographic, and genetic factors. And we're really interested in particular in the crosstalk between the microbial and genetic factors and in its relationship to the immune system, and understanding how that interplay contributes to immunity and disease outcomes. And so there's no question that the microbiome affects all aspects of health. And there's been a lot of progress in this field. But what about viruses? How do viruses contribute to health and immunity? And collectively, viruses collectively known as the virome, how do they contribute to health and immunity? And so, and this is an area that I've been interested in for a long time now. And so the human virome is a collection of viruses that most of us harbor. And this consists of many herpes viruses that are commonly acquired during childhood, like EBV and CMV and HHV6. And these are highly prevalent in the population. And in fact, a recent paper showed that viruses like EBV and HHV68 are highly prevalent in tissues throughout the body. And this is indicated here on the right. But sort of unlike other viruses that you may not see on this list, like influenza, norovirus, or even coronaviruses, these particular viruses are able to establish a lifelong infection without causing much harm to the host. And that means that there's this persistent and chronic interaction between viruses and the immune system. And this could really influence disease outcomes by shaping our immune response at baseline. And this has been beautiful work been done by pioneers like Skip Version and others in the field who have really shown the impact of the virome on our immune system. And at the top of this list is our favorite, which are the endogenous retroviruses, and these are genome encoded sequences that are present in all human genomes. And so what are ERVs? Endogenous retroviruses are relics of ancient retroviral integration events. And that means that they originated from infection by exogenous retroviruses, much like HIV. And during infection, the viral RNA reverse transcribed, and the proviral DNA integrated into the host genome. And those proviral sequences that integrated into the germline, that were stably inherited, eventually became fixed in the primate genome. And these sequences are now endogenous or endogenous retroviruses. And these sequences are thought to date back to integration events anywhere between 0.5 to 40 million years ago. There are some endogenous retroviruses like the ERV-K family or the HML2 family of ERVs, which is a more recent integrant to the Homo sapien lineage. And I'll come back to that a little bit later. And so ERV sequences collectively make up roughly 8% of the human genome and 10% of the mouse genome. And these sequences are present in most eukaryotic genomes. So that's quite a lot of real estate, right? Given that canonical protein coding sequences make up only 2% of the genome. And this then really begs the question of how these viral sequences affect physiological processes, including immune functions. And ERVs have been implicated in many diseases, including neurodegenerative diseases, autoimmune diseases, and even cancer. But really, there is still a major gap that remains in really understanding the mechanistic link between dysregulated ERVs, the immune system, the immunity, and disease outcomes. And this is really where our interest lies. And so today, I thought I would give you a quick overview of endogenous retroviruses for those of you who are not really familiar with this field, and an overview of sort of my new research program here at UBC. I'd like to highlight some of the work that was done mostly while I was still at Yale in Akiko Iwasaki's lab, and end with some highlights for directions and some new data from our lab. Okay, so digging a bit deeper into what endogenous retroviruses are. So as I mentioned earlier, ERV sequences originated from proviral sequences upon retroviral infection. And initially, these sequences were likely full-length proviral sequence that contained sort of all the viral open reading frames that were required for viral replication. And these were flanked by both the 5' and the 3' long terminal repeats, or LTR sequences. However, over the course of evolution, these sequences have acquired a lot of mutations and deletions, which have resulted in the generation of fragments of internal sequences, or a generation of what we call solo LTRs, which resulted from the recombination of the 5' and 3' long terminal repeat sequences. And really, it's estimated that the majority of these sequences take these truncated forms, but there's still a significant fraction that has retained the proviral structure, including intact open reading frames. And so what do these sequences do? So there are different types of ERV sequences, as I mentioned, in the genome. And so what's really been very well established at this point is that solo LTRs are really good cis regulators of gene expression. And they can serve as alternative promoters or enhancers to regulate expression of nearby genes. And so here's one beautiful example from Cedric Fauchot's lab a while ago, where they showed that a solo LTR element called the miR41 LTR that is upstream of AIM2 gene is actually a promoter to regulate expression of AIM2 in an interferon gamma dependent manner. And so if you delete this miR41 element, you lose this interferon gamma responsive AIM2 expression. And they found that this is due to this STAT1 binding consensus binding site that is present in the miR41 sequence. And so there's several examples like this and really illustrating the nature of these elements as sort of regulators of gene expression. On the other hand, proviral ERV sequences can generate viral proteins like GAG, polymerase, and envelope, and these can modulate the host similarly to other viral products. And so as a viral immunologist, we're very much interested in particularly into some of the interactions between some of these ERVs that still encode viral open reading frames and the immune system. And so we know that generally ERV expression is under the control of epigenetic silencing. And so, however, there are many, in various contexts, there's a release of this epigenetic silencing, and this can lead to expression of ERVs. And this can happen in various disease contexts. We also know that viruses and even microbial cues can also lead to activation of these endogenous retroviruses. And so what are some of the consequences of this ERV dysregulation and immunity, and what do we know about them so far? And so we know that there's an emerging area really suggesting that these endogenous retroviruses serve as ligands for innate sensors to turn on interferon signaling. We also know that viral proteins that are encoded by these endogenous retroviruses can form extracellular vesicles, or these proteins themselves can turn on cell surface TLRs like TLR4 and TLR2 to activate pro-inflammatory cytokines. We also know that there's immune surveillance of these from the adaptive immune system, whereby there are antibodies that are generated against these endogenous retroviral proteins. And furthermore, there's been a lot of work now showing, especially in cancer, generation of these peptides that are derived from these endogenous retroviruses, which can then actually be sensed by T cells in the system. In the past, there's been studies that have also shown some superantigen potential of these endogenous retroviral proteins to activate T cells. And so, but given how how many ERVs are encoded in the genome, where we really only likely scratched the surface in terms of their impacts on the immune system, and this is sort of where we're hoping to make further contributions in. And so my lab is really interested in three major areas. One major area is in advancing our understanding of ERV sequences, when they're expressed, in what context, and really trying to identify the translated products of these ERV transcripts, both using transcriptomics and proteomics. In the second arm, in the second arm of my research program, we're really interested in understanding how these endogenous retroviruses actually affect immune cell functionality and immune surveillance, both cell intrinsically and cell extrinsically. And then lastly, we want to probe then how these interactions between these ERVs and the cellular immune system can both leads to beneficial effects like pathogen clearance and detrimental effects like promotion of chronic or contribution to chronic inflammation in the case for autoimmune diseases. And really, ultimately, we're hoping that by studying these interactions that we are able to reveal some novel pathways of immune modulation that can explain individual differences in disease and potentially can be targeted for therapeutic purposes. And so how do we do that? So our approach has been really reliant on starting with clinical samples or clinical data, and then use them really to quantify ERV transcriptome using our favorite pipeline, which is ERVmap. And really we use this pipeline to then narrow down some of the ERVs that we're really interested in studying in terms of its relationship to the immune system, and dissecting sort of the functional impacts of these endogenous retroviruses on various immune cell types. And we use the study to also further use animal models to investigate some of these relationships and probe the role of these in disease outcomes. And so I'll highlight some of the work in this area that I've worked on in the past. And so in the first story, I'll highlight the work that we've done that was on the role of endogenous retroviruses in systemic lupus erythematosus. And so systemic lupus erythematosus, or SLE, is an autoimmune disease that affects systemically various organs throughout the body. And really, this affects a large population of individuals worldwide. There are over 5 million people worldwide who are affected with lupus. And 90% of individuals affected by lupus are female. So it's a very female dominant disease. There's higher prevalence in African Americans, Asian, and Hispanics. And really the disease manifestation is highly heterogeneous, which makes it very hard to treat. And due to this complication, really, we've only had 2 FDA-approved biologics, belimumab, which is an anti-BAFF monoclonal antibody, which was approved in 2011, and anifrolumab, which is an anti-IFNAR 1 monoclonal antibody, which was approved in 2021. And so, there's been a long-standing interest in trying to dissect whether there's a viral etiology of disease. And in fact, endogenous retroviruses have been implicated in lupus for a very long time. And so prior even to our work, there were studies in SLE patients that had shown that endogenous immune complexes containing these endogenous retroviruses are present in the serum. There have been studies that have shown antibodies specifically against an ERV protein, the GAG protein, that are found in the serum of SLE patients. And furthermore, there have been T cells identified that are specific to an ERV protein, HRES1. And these have sort of been detected in the past, but really we didn't have information on how that actually contributed to disease or the mechanism of contribution to disease. We also, on the other hand, knew that in many of the mouse models of lupus, including some of these listed here, all of these mouse models, in fact, have highly elevated GP70 or ERV envelope protein expression in the serum. And so you can find a lot of these GP70 in the serum of many of these mice. And you can also find, furthermore, that these immune complexes can deposit in the kidney, suggesting that there's an involvement of, suggesting that these can actually contribute to inflammation and disease in this case. And so with this in mind, we were really interested in understanding, more broadly, whether ERVs are dysregulated in SLE disease, whether ERV expression actually correlates with features of clinical disease, whether ERV, whether these ERV antigens are actually targeted by autoantibodies, similar to other autoantigens in lupus, and if so, what happens to what's the consequence of this reactivity is sort of what we were interested in probing. And so, to begin with, to really understand sort of at the global transcriptome level how these ERVs are dysregulated, we utilized this pipeline that we had developed back in 2018 called ERVmap. And this ERVmap really allows us to dissect and quantify the amount of proviral ERVs that are expressed in transcriptome data. And so just briefly, this pipeline really includes a database of proviral sequences that have a relatively intact ERV sequence. And in addition to that, we have come up with some stringent pipelines to be able to grab uniquely mapped reads to have more specificity in the assignment of these reads. And at the time, we had also generated a web tool to go along with this. And so using this ERV map, we really wanted to dissect, again, how these ERVs are dysregulated in lupus pathogenesis. And so this was work in collaboration with Dr. Insoo Kang, who's a rheumatologist at Yale, and 2 students that I had worked with when I was still at Yale, Arvind Venkataraman and Toshiko Jayawekar. And so using some of the data that we had obtained from PBMCs from lupus patients from the Yale cohort, as well as published data that we had obtained from Genentech, we found that ERVs, in fact, are highly upregulated in SLE patients, as you can see here in this cohort on the left and also on the cohort on the right, where SLE patients are really expressing a lot more of these endogenous retroviruses compared to healthy controls. And this really expanded sort of our repertoire of endogenous retroviruses to really study in this disease, because previous to this, we had only known 2 distinct ERV loci that were associated with this disease. And so given that in mind, we first asked whether these endogenous retroviruses or expression of these endogenous retroviruses correlated with clinical disease. And so we had from one of these cohorts data on the level of autoantibodies that are present in these patients. And of course, autoantibodies are a hallmark of SLE pathogenesis. And so looking at the correlation between SLE autoantibodies and ERV transcripts, we can see a significant and a positive correlation between endogenous retroviruses and autoantibody levels. I'm just having trouble finding my arrow here. Okay, here we go. All right. And so we can, we then asked, more specifically, whether a certain, a specific ERV loci was involved in lupus pathogenesis. And for this, I'm going to go back to what I had mentioned earlier in the background slides, where I mentioned that there are some ERVs that are actually very Homo sapiens specific, that are recent integrants. And so these are the ERV-K family or HNL2 family of ERV sequences. And because they're more recent integrants, integrated sequences, they're more intact compared to some of the other ones, and they've been actually implicated in a number of diseases. And so in our ERVmap database, we've pulled out 87 ERV-K family member sequences, 12 of which at the time encoded what we had known encoded envelope proteins sequences. And so using this, we specifically looked to see whether these ERV-K envelope proteins were dysregulated in lupus pathogenesis. And in fact, we found 4 of these ERV-K loci that encoding envelope sequences that are significantly upregulated in lupus patients. And these are ERV-K102, ERV-K10, ERV-K106, and ERV-K115. And so these envelope encoding ERV-K sequences were found to be significantly upregulated in SLE patients. And looking at these further, we narrow down to sort of our focus to this highly expressed ERV-K locus, which is the ERV-K102 locus, again, encoding an intact envelope sequence. All right, so this ERV-K102 sequence, when we dug a little bit deeper into its characteristics in terms of its expression, we found that it is actually significantly upregulated in female patients compared to male patients, which is consistent with sort of the female dominant disease in lupus pathogenesis. We also found that ERV-K102 expression correlates with the interferon signature that we see typically in SLE patients. So those patients that had a higher interferon metric signature or ISM score had a higher expression of ERV-K102 compared to those patients who had a lower interferon signature metric. And so with this in mind, we were really curious to know whether these ERV-K, the envelope encoding ERV-K102 sequences can be, and proteins can be targeted by autoantibodies in lupus pathogenesis, and could that actually then contribute to, can that contribute to some inflammatory consequences of disease? And so we generated a recombinant envelope, K102 envelope, and used that to probe the titers of anti-ERV-K102 envelope IgG initially, and also different subclasses of antibody in lupus patients. And what we found, a bit disappointment, but initially we were slightly disappointed that that both actually, in fact, healthy and SLE individuals express similar amounts of, or possess similar amounts of anti-ERV-K102 antibodies, both at total IgG levels and in different subclasses as well. But what we found when we looked a little bit deeper was that despite the fact that the total titer of SLE and healthy ERV-K102 IgG were similar, they seemed to correlate differently in terms of in terms of inflammation and inflammatory signatures and lupus pathogenesis. So here I'm showing you the titer of anti-ERV-K102 on the x-axis versus expression of some of the ISGs that are highly expressed in lupus patients. And what we can see is that there's a really significant and positive correlation between the amount of ERV-K102 IgG that are present in lupus patients and the extent to which they express these ISGs in the blood. And so then we asked, despite the fact that the total overall titers were different, could these IgGs actually be functionally different? And so to really probe this question, we collaborated with Galit Alter and her former postdoc, Bonnie Gunn, and try to understand sort of the functionality of these antibodies using their system serology assays that they had already set in place and had been is an expert in. And so using some of their assays, what we found was that there was one particular phenotype that was really striking, which is that when we make these, when we generate immune complexes with either healthy serum or SLE serum with the ERV-K102 envelope protein and feed them to neutrophils to assess antibody-dependent phagocytosis by flow cytometry, what we found was that when you take serum from SLE patients and incubate them with these ERV-K102 envelope proteins, you see an enhanced level of antibody-dependent neutrophil phagocytosis only when you make these complexes with SLE serum, but not when you make these complexes with healthy serum. And this can be seen with total plasma. And you can also see this even more pronounced when you purify the IgG from the plasma and perform a similar experiment. In contrast, we don't see this enhancement of antibody-dependent neutrophil phagocytosis against an irrelevant antigen or a common antigen like influenza HA or tetanus toxin, which most people should be vaccinated against. And so this was really interesting to us, and we probed, whether the functionality, how these antibodies were distinct, and try to characterize this a bit more. And so one of the experiments that we did was to assess whether the enhanced antibody-dependent neutrophil phagocytosis was due to the fact that these antibodies from these SLE patients can have a higher binding to Fc gamma receptors. And so again, in collaboration with Galit and Bonnie, we did this Fc gamma receptor binding assay, and what we found was, in fact, this the binding of these immune complexes, these ERV-K102 immune complexes with Fc gamma receptor 3b, was what correlated the most with the enhancement in antibody-dependent neutrophil phagocytosis score. And if you actually block Fc gamma receptor 3b on neutrophils prior to incubating them with these immune complexes, you then lose this enhanced antibody-dependent phagocytosis that we saw before, suggesting that these somehow these SLE antibodies against ERV-K102 IgG are leading to immune complex phagocytosis by neutrophils through Fc gamma receptor 3b. And furthermore, what we found was that when you feed these immune complexes to healthy neutrophils, that immune complexes really generated with SLE antibodies and ERV-K102 antigen leads to full activation of neutrophils and generation of these neutrophil extracellular traps, or nets, as shown by staining with citrullinated H3 and neutrophil elastase here. And you can only see this when these complexes are generated with the SLE plasma, but you don't see that with the complexes generated with healthy plasma or SLE plasma alone, and really suggesting that these antibodies are quite distinct in SLE patients compared to healthy donors, despite the fact that we found the levels to be quite similar. And netosis is really a hallmark of SLE disease, and in fact that promotes this positive feedback look of type 1 interferons signaling through sort of generation of immune complexes against these nucleic acids that are now generated from these nets. And so what we think is potentially happening is that these ERVs may contribute to this positive feedback loop of inflammation through contributing to neutrophil activation in this manner. And so just to quickly summarize this, this part, what we had shown is that in healthy individuals, they compared to SLE individuals, SLE patients, they express low levels of ERVs. They do have anti-ERV-K antibodies, ERV-K IgG, but they don't correlate with inflammation or ISGs. They don't seem to have ability to be able to promote neutrophil, antibody-dependent neutrophil phagocytosis, and they also don't form, and neutrophils do not form nets when they see these immune complexes. In contrast, in SLE individuals, SLE patients, they express much higher levels of endogenous retroviruses. They also have anti-ERV-K IgG, and these anti-ERV-K envelope IgG seem to be quite distinct from the healthy donors, that they correlate with, in fact, the levels at the individual basis correlate positively with ISG expression. These antibodies lead to enhanced antibody-dependent neutrophil phagocytosis of immune complexes, and these lists also leads to netosis. And so really there are a lot of remaining questions in this area, and some of which we're starting to address in my lab at UBC. And so some of the questions that are still remaining include, for example, how are these anti-ERV-K IgGs distinct between healthy and SLE? I've kind of shown you that they're functionally distinct, but what is the difference? What are some features that make them distinct? We still don't quite know what those are. It's really interesting that these anti-ERV-K antibodies are also present in healthy individuals, and yet they don't seem to cause any inflammation. And so what are some of the inflammatory conditions that actually cause these antibodies to become now pathogenic in the context of SLE? And therapeutically, you know, would it be worthwhile to block the inflammatory anti-ERV-K or pathogenic anti-ERV-K antibodies from sort of causing further inflammation? And sort of in addition and in parallel to all of this, I showed you from the RNA sequencing data that there are a lot of other ERVs that are dysregulated in SLE. We've really only focused on one, ERV-K102, but, you know, beyond that, what are some other ERV proteins that are targeted by autoantibodies, and how does that contribute to inflammation in lupus? And these are some of the areas that we're still trying to address here. And so shifting gears a little bit, I would like to share more recent work that we recently published on showing how these endogenous retroviruses now can also confer protection against an exogenous virus, and that it can also not, you know, serve as sort of have a beneficial effect on our immune system. And so antiviral immunity by ERVs have been shown in several models in the past. And so murine leukemia virus, or the mouse endogenous retroviruses, have been shown to be able to block entry of exogenous viruses, retroviruses. It's also recent paper has also shown that these retroviruses can actually bind to the receptor ASC2, and these human endogenous retrovirus envelope protein can, through binding to ASC2, can block, again, an exogenous virus from entering the cell. And we also know from sort of the HIV field that these endogenous retroviruses, due to its similarity with HIV proteins, it can actually compete with HIV proteins like HIV GAG and prevent further replication of HIV. So here, if you overexpress ERV-K GAG in the context of HIV infection, then you actually see less HIV RT activity coming out from these cells. And so there's been suggestions that these ERVs can also, and there's been evidence showing that these ERVs can also mediate antiviral immunity. But, we didn't really quite know whether that can actually modulate the host, and if that can promote a broader sort of antiviral response against exogenous retroviruses. And so this is sort of where we were interested in. And so this is the work that was that I worked with Tengyang Mao and Radisha Jayavikram, again, still while I was in Akiko's lab, and recently finished this project here at UBC, where we're really trying to, where we really ask the question of how do herbs affect the host, and how does that in turn impact exogenous viral replication in vivo. And so this work was using herpes simplex virus, or HSV2, as a model pathogen to really answer this question. And so just a quick background on HSV2, or herpes simplex virus 2. It is a DNA virus that affects, over 400 million individuals worldwide. So there's a really significant health burden across the globe. Roughly 60% of the neonatal infection, third vertical transmission, is fatal without treatment, and we still don't have a vaccine against this. And HSV2 can also be a contributing factor for other viruses like HIV, HCV, and bacterial vaginosis through damaging the epithelial, the layer of the genital tract. And also there's been some increase in acyclovir-resistant HSV2. And so there's still quite a need for understanding potential therapeutic therapeutic strategies to target HSV2. And so HSV2 is a infection that occurs in the female reproductive tract, or the FRT. And really what happens is that HSV2 affects the vaginal epithelium, as indicated here, which leads to the recruitment of innate and adaptive immune cells at the site. And the HSV2 can also establish latency in the dorsal root ganglion. And then over time, it can also reactivate and cause epithelial damage at the site of reactivation. And so again, this is a persistent infection, and it can cause lifelong symptoms and discomfort in individuals who are affected. And so in the context of ERVs, so how do we study the role of ERVs in HSV2 infection and disease? And this really comes from the work that was done by George Cassiotis and Stephen Bauer. George Cassiotis at the Francis Crick Institute had shown back in 2012 that there is a resurgence of infectious ERVs in immunocompromised strains of mice. And this is a really exciting piece of work that really showed that in certain commonly used strains of immunocompromised strains of mice, like RAG1 knockouts, even MyD88 and TLR7 knockouts, there's this huge resurgence of these endogenous retroviruses, or these endogenous murine leukemia viruses that are expressed in these mice. And in fact, the mouse genome encodes, a large number of proviral sequences, and in particular, kind of focusing in on the TLR7 knockout mice that express high levels of endogenous retroviruses. George Cassiotis's work, and we've subsequently did a similar study to show that in the TLR7 knockout mice, the infectious ERVs that are kind of re-emerging from these mice are a recombination between this two proviral loci, EMV2, which is ecotropic endogenous retroviruses, and XMV43, which is a xenotropic endogenous retroviruses. And complementation between these two proviral sequences leads to the resurgence of infectious ERVs in these mice. And so we've shown that, in fact, in these mice, in these TLR7 deficient mice, these infectious ERVs are systemically elevated. And so we can actually measure that by co-culturing DFJ8 cells, or avian cells, with leukocytes that express high levels of endogenous retroviruses obtained from, either B6 or TLR7 knockout mice. And then by co-culturing these for 5 to 7 days, you're actually going to be able to see DFJ8s expressing the envelope envelope protein for these endogenous retroviruses, indicating an infection. And so with this assay, we've been able to show that systemically, these endogenous retroviruses are highly infectious endogenous retroviruses are expressed systemically, both in various tissues and also in the vaginal wash. And so kind of using these mice, we probed initially, whether these mice have different susceptibility to HSV2. And so to do this, we intravaginally infect mice with herpes simplex virus 2 after pretreatment with Depo-Provera, which sinks them in the diestrus cycle. And so when we did this experiment, what we found was that, in fact, the TLR7 knockout mice that have high levels of endogenous retroviruses are more resistant to HSV2 compared to wild type mice. And this was observed both in disease score and also in survival. And we see that at day 3, there's also a lower amount of viral titers in the vaginal washes of these mice as well. And so this was very exciting to us that we were able to see this difference in HSV2 susceptibility, and really to be able to know whether this was actually due to different levels of endogenous retroviruses between these two strains. Here we're gonna, and this is sort of showing you the expression of these endogenous retroviruses in the vaginal tract. And so this is an immunostaining for endogenous retrovirus envelope, and using a monoclonal antibody called 8325, and we're able to see this nice expression of these ERV envelope proteins. And this is sort of where you can see HSV2. This is staining for anti-GB and HSV2. You can see that this HSV2 is, in fact, penetrating through this vaginal epithelium in both wild type and TLR7 knockout. But what you can see is sort of this the lack of spread in the TLR7 knockout in the vaginal epithelium compared to the wild type. And this might be sort of the reason for why we see lower disease score in these mice. And so kind of going back to, again, to my original thought of, so how do we know that this is actually dependent, this phenotype is actually dependent on ERVs? Well, we took 2 approaches to really test this. And one approach was to isolate these endogenous retroviruses from the TLR7 knockout mice and exogenously feed these virions to wild type mice that are susceptible to HSV2. And so if you pre-treat wild type mice with these endogenous retroviruses prior to HSV2 infection, and then monitor for disease, what we saw was that there is some delay in disease, in HSV2 disease development, and also increase in survival of these mice that are pre-treated with endogenous retroviruses. And we can also see something similar in an even more susceptible strain, an IFNAR knockout mice. And if you, again, pre-treat IFNAR knockout mice with these endogenous retroviral particles prior to HSV2 infection, again, what we see is that we see this delay in HSV2 disease and an increase in survival, really suggesting that these ERVs are sufficient to promote delay in HSV2 disease. And this seems to not require the interferon signaling pathway, as again, this is seen in an IFNAR knockout mice as well. And to kind of test this genetically, we then further deleted the EMV2 locus, which again is responsible for generating these infectious ERVs in the TLR7 knockout mice. And so with the help of Bill Philbrook at Yale, we generated these EMV2 TLR7 double knockout mice, where we deleted the entire EMV2 locus. And when we, and as you can see in the RNA sequencing data, we no longer in the EMV2 double knockout vaginal tissue, we no longer see EMV2 and XMV43 compared to the TLR7 knockout mice. And you can also confirm deletion of the EMV2 by surface staining for the ERV envelope protein on leukocytes like splenocytes, where there's enhanced expression of the ERV envelope protein in TLR7 knockout mice, but this is completely gone in the EMV2 TLR7 knockout mice, suggesting that we really deleted this locus. And so using these mice, again, we infected them with HSV2. And what we found was that, in fact, the resistance that we were seeing against HSV2 in the TLR7 knockout mice is now gone in the EMV2 TLR7 double knockout mice. And again, this is reflected both in the disease score and survival. And so why is that? What is happening and what is sort of the mechanism here? So we're still trying to probe the mechanism of this protection here in my lab, but there's a few things that we have done to kind of rule things out, actually. And so, for example, we did a vaginal, we performed an RNA sequencing on total vaginal tissue to look for any differentially regulated genes in these EMV2 or in the TLR7 knockout mice that are absent in the EMV2 TLR7 knockout mice. So an EMV2 dependent antiviral signature is sort of what we were probing for. And in fact, what we found were a lot of genes sort of that are EMV2 dependent that are upregulated in the TLR7 knockout vaginal tissue that actually involved quite a lot of sort of extracellular matrix organization, which was surprising that there were not a ton of immune genes or canonical antiviral genes that are involved in this process. And so, to further confirm this sort of the absence of canonical antiviral signature here, we also specifically looked at some of the ISGs that have been known to be involved in protection against HSV2. And again, we didn't really see any difference between wild type and TLR7 knockout mice compared to the EMV2 single knockouts and the EMV2 TLR7 double knockout mice as shown here. We also didn't see enhanced recruitment of dendritic cells, which are also really important for mounting that antiviral response in these mice. And so again, we're still not quite, we don't have all the answers here yet, but it didn't seem to be part of a canonical sort of interferon dependent antiviral response that was promoting this protection against HSV2. And so recently we've been trying to probe this a little bit more. In one, for example, one experiment we had done recently was try to understand whether these endogenous viral particles and the ability to bind to the vaginal epithelium was required, or even viral replication was required for this protection. And so to test this, we either neutralized these endogenous retroviruses with a monoclonal antibody against the envelope 8325, or UV irradiated the virus prior to injecting wild type mice with these particles, and then infected them with HSV2 to look for survival. And what we found was that neutralization of these virions with the monoclonal antibody 8325 actually led to the loss of protection that we see with these viral particles, but it didn't require replication. So UV irradiated virus can still promote this delay in this HSV2 disease. And so we're currently sort of pursuing this idea that this binding between these endogenous retroviral particles and the vaginal epithelium is really required to mount this this antiviral response that is interferon independent. And one experiment that this is a project that one of my graduate students, Austin Guild, is working on, where he recently tried to look for interacting partners between these endogenous retroviruses and the vaginal epithelium. And so we took wild type B6 mice and vaginal tissue lysates from these, incubated with these endogenous retroviruses, and immunoprecipitated using this monoclonal antibody 83825 against the ERV envelope protein. And when we did this co-immunoprecipitation assay, we found these really two striking bands. These are two replicates, these bands that were really prominent in these co-IP lanes that were not present in the antibody alone, or the ERV virion or ERV virion alone lanes. And so we performed a mass spectrometry analysis on these bands to try to identify what they were. And we pulled out a number of really interesting candidates that were interacting with these endogenous retroviruses. Of course, the endogenous the retroviral ecotropic murine leukemia antivirus protein was a top hit, which is a good sign that our immunoprecipitation was working. And sort of the second hits and the subsequent hits were really interesting. One being albumin, which is the really abundant protein, the most abundant protein in many of the bodily secretions. And albumin is sort of known to be a carrier protein to allow for uptake of various materials through various cells and epithelial cells in particular. And so we're sort of pursuing at the moment this idea that along with some of these other hits, that potentially these endogenous retroviruses are being taken up by the vaginal epithelium by sort of conjugating with albumin in the lumen, and that's kind of leading to this promotion of an antiviral response against herpes simplex virus. And so to kind of summarize this, again, the second project, we showed that in TLR7 knockout mice that express high levels of endogenous retroviral proteins, they're actually protected against HSV2 infection compared to wild type or the EMV2 TLR7 double knockout mice. And it seems to be interferon independent. So we can see this protection in the IFNAR knockout mice, and we don't really see a heightened interferon response in these mice. And so potentially there's an interesting alternative sensing mechanism of these ERVs that may be promoting an antiviral response. And so some of the questions that we're currently trying to address in my lab is, can we actually boost this ERV-mediated antiviral response? I showed you that there's some delay in HSV2 disease, but can we even boost that even more by enhancing potentially the interaction between these ERVs and the vaginal epithelium through maybe carrier proteins like albumin? And this is something that we're currently pursuing. And what we ultimately want to know is sort of therapeutically, is it possible to identify minimal components of these endogenous retroviruses that can alone be sufficient to boost an antiviral response again in the genital tract? And so endogenous retroviral sequences are not the same between human and mice. And so there's not sequence homology between mouse and human. However, there are viral proteins that are like envelope proteins that are functionally similar between mouse and human. And if we can sort of narrow down the identity of the endogenous retroviruses in mice that are promoting this antiviral response, we're interested in now carrying that forward and finding potential functional homologies in human as well. And sort of long term, we're really interested in understanding how these ERVs are also protected, potentially if they're protective at other mucosal sites as well. And so I hope that I've kind of given you sort of an overview of endogenous retroviruses and how they can activate or how they can interact with the immune system, both for beneficial purposes and also for detrimental purposes. And so, and detrimental scenarios. And so, as there are a lot of different ways in which endogenous retroviruses can potentiate inflammation and activate the immune system. And sort of we're just getting started in terms of the interaction and the intersection between ERVs and the immune system. And really, I think this is an exciting time to be in this area with all the tools that we now have. Can studying a lot of these interactions allow us to really give us novel insights into fundamental processes in immunology, as well as further understand how these interactions may be contributing to heterogeneity in disease. And so we're hoping in the coming years that we can contribute more to this field. And so lastly, I'd like to acknowledge my current lab here at University of British Columbia. I have 4 graduate students and a part-time technician in the lab. We've had some undergraduate students that have worked with us in the summer. And I've also like to give a big thank you to the Iwasaki lab at Yale, many of whom I acknowledged throughout my talk, who have been really instrumental in getting a lot of this work out and allowing me to sort of build a really strong foundation for future work here, and acknowledging my funding sources here as well. Thank you, and I'm happy to take any questions.
Thanks so much, Maria. That was really fantastic. There are a couple of questions on YouTube that I'll read. One is from Vivian Liang. Really interesting talk. Does the anti-ARV antibodies cross-react with non-ARV human antigens? And two, given what you observed is envelope antigens, does it mean that active viral replication is needed?
So I guess maybe I'll tackle maybe the second question first. We don't. So I think clarify, there haven't been any infectious ERV variants that have been detected in humans. There are extracellular vesicles that are coded in envelope proteins that are actually present in many that have been detected in many scenarios. So those can potentially be targets of autoantibodies as well, or antibodies. But replication competent ERVs have not been found in humans as of date. In terms of the specificity and sort of the ERV antibodies that we found in SLE patients, we haven't actually probed that more experimentally. I don't have an answer. However, there are, class switch antibodies against these ERVK proteins. We find our anti-ERVK 102, IgG 1, 2, 3, and so they are, class switch antibodies that we find, and whether that cross reacts with other proteins, we don't have that answer yet, but we don't know.
Thank you. The next question is from Innate Adaptive. Is the transcription of ERVs age dependent? Do you think differential activation of ERVs could shape infection susceptibility in young versus old people?
Yeah, this is a fascinating area. And certainly due to epigenetic changes that occurs through aging, this is a really, tantalizing hypothesis that aging can induce expression of dysregulation of ERVs, and that can potentially mediate a lot of these consequences, right? We did look at the correlation between age and ERV expression in lupus patients, and we didn't see any correlation that was obvious to us in that patient cohort. That being said, that's not to say that this dysregulation, age-dependent potential dysregulation of ERVs is not happening in other contexts, but we did not see that in the lupus patients. But interesting.
Thank you. Next question is from Debbie Lebesma. Do ERVs tend to be preferentially expressed in some tissue over others?
Yeah, so there's actually been now a few studies that have looked at the done a transcriptomic analysis of various tissues and found, that there's specificity in terms of the type of ERVs that are expressed in different tissues. I don't know if are there any tissues that don't express, that are not able to express endogenous retrovirus? To a certain extent, most tissues are able to express to varying degrees one or more of these endogenous retroviruses. I think, we still have to do a lot more of these types of analysis to be able to really, conclusively be able to define, right, which ERVs are expressed in what context and what cell types. And I think this is an area that we're hoping to also contribute in because there are many, many ERV sequences in the human genome. And really when we're doing these studies, we're really focusing on the highly expressed ones that we can pick up in our transcriptomic assay. So there is some bias there. But yeah, that variety of tissues have been shown to express endogenous retroviruses. Simple answer.
Awesome. Thank you. And it looks like the last question is from Cecile. Do you think ERVs are immunogenic per se? Can they trigger a T cell immune response?
So I guess there are, I would say that, in the context of cancer, a lot of these ERVs are being studied as a potential source of neoantigen. And really there have been T cells, that have been identified in the context of cancer that are specific to endogenous retroviral peptides, right? And so in that sense, yes. And whether that is happening in other contexts and how often that happens and what is actually triggering that, what are all the signals that are required for this T cell response against these endogenous retroviral peptides, I think there's still a lot more to be done. I think we're only scratching the surface in these areas.
Thank you so much. That was beautiful. Thank you.
Thank you.
We'll end the
Thanks for
live stream.
listening, everybody.